5 or 6 years ago, maybe it's a bit longer, we weren't sure the therapeutics initiative would survive. And Dr. Barbara Mintzes and I met with David Patrick, who was then the director of the School of Population and Public Health in BC. He was a strong supporter of ours because he understood the value of conflict-free science. And this is a chance to thank him for that, amongst other things. Now, before I introduce him properly, I need to acknowledge we are meeting on the unceded ancestral territories of the Musqueam, the Squamish, and the Tsleil-Waututh peoples here in BC. Where I'm coming to you from right now on 12th Avenue, I'm almost right at the old territorial boundary of the Squamish to the north and the Musqueam to the south. So we appreciate their hospitality to us to be here. And I might add that we probably all thank God that we're in Canada now, not some other parts of the world. David Patrick is he's an infectious disease physician and an epidemiologist. He is a full professor at UBC. He is particularly interested in emerging infectious diseases and in the role of antibiotics and wise antimicrobial stewardship. He chairs BC's COVID-19 Strategic Research Committee. He publishes widely, including recently in the Lancet Respiratory Medicine on a topic similar to what we're about to hear about tonight. And even more important, I don't know if you realize this, David, but you have a namesake who shows up in your PubMed search, who's a clinical pharmacologist at Vanderbilt University in Tennessee, which is an excuse if there are any Americans listening tonight, we are thinking of you from up here in Canada. And take it away, David. Nice to have you with us.
Thanks, Tom. I think of that David fondly whenever I do a PubMed search to put my annual report together. So I'm going to share my screen here, folks. And we are going to put this on slideshow. Okay, first of all, I'd like to thank Dr. Perry and the Therapeutics Initiative for having us here. I'm going to talk about a topic that we sort of began to discuss in the literature just as the current COVID pandemic was breaking. And in a way, this was unfortunate timing because we really think that if our hypotheses prove correct in the long run, they point a way to making a dent in another pandemic. However, I think a hypothesis needs to be attacked from all angles. And the one thing I know about the TI and the people who follow along with it is that they're a great bunch of skeptics. So I look forward to having this conversation. We've been working hard to explore this area, but we really want to have some good hard questions. The other thing I'll say as a disclaimer starting out is I'm representing a big group and I am the infectious disease clinician and epidemiologist. So if I don't give you quite enough depth on the explanation of the genomics, for example, I'll apologize in advance, but I'll tell you a little bit more about our groups here. And just got to get this thing. Yeah, there we are. So the first thing to note is that I'm representing a big group. You should know about our funding. The funding for the population level analyses comes ultimately from British Columbia Ministry of Health Pharmaceutical Services branch that funds the community stewardship program that I run. The child study, which is a prospective cohort of infants in Canada, is funded mostly by CIHR, but also by Allergen and the Networks of Centers of Excellence, who provided core funding. And there were other funders that came in there. There's a list of people who contributed to this work on the right-hand side of the page. And I just want to point out that it was an important collaboration between my group at BC CDC, Hind Sabihi, Darlene Dye, and Stuart Turvey at the Children's Hospital, and Brett Finley's lab at UBC Michael Smith Center. Now, the first thing I think all of us take for granted is that childhood's most prevalent chronic disease is asthma. And we sort of treat it as if that's the way it's always been, but that's not actually the way things have been historically with asthma and other things. Oh, brother. Okay. So the interesting thing is that while the prevalence is about 8% in North America now, and it's important to note that the developed world prevalence essentially doubled toward the end of the 20th century. And over on the right-hand side of the page, you can see the increase in asthma prevalence in the United States from 1980 through to about 2010, after which it was felt to have more or less plateaued. But one thing that strikes me as funny as an infectious disease epidemiologist, or if you've been watching the COVID epidemic, is that prevalence only tells part of the story of an epidemic. If you want to know what's happening now or what's happening dynamically, you need to look at incidence. And we don't do that often enough with chronic diseases. Incidence is indeed falling in many parts of the world. It's been declining in parts of the USA, Canada, and the UK over the last 10 years. On the right-hand side of the page is CIHI data, which has shown that hospitalizations for asthma actually plummeted 50% in a decade across Canada. And so you have to ask what exactly is going on. If you are involved in environmental health, it's fair enough for you to say, okay, air quality is improving. We've done a good job of getting cars re-engineered. Fewer people are smoking inside. That's got to be the explanation. That's a great hypothesis. If you are a clinician who's been involved with asthma therapeutics, you may well say, okay, pre-hospital care is way better, and that's what's driving down hospitalizations. But if it's true that incidence is declining, there's a number of us who are also asking, what about the environment within the human body, and specifically the impact of the microbiota? Now, this may seem a little bit from left field for many people, but I wanted to give you a little bit of a background of what's out there in the literature, if you've not checked this lately, in terms of antibiotics and the infant microbiome. The first thing is that if you look at a number of observational studies of cross-sectional or cohort design, you can usually see a higher odds ratio or relative risk for asthma associated with cesarean section, but especially cesarean section without labor, with a failure to bottle feed, and with infant receipt of antibiotics or maternal receipt. And by and large, if you take a look at meta-analyses in the literature, the relative risk sits around 1.5, with estimates ranging from 1.2 to 2.0. In early analysis of the child cohort study in Canada, and I'll tell you more about that study in some detail, it was discovered that the absence of several key organisms in the infant gut was associated with a higher risk of atopic tendency and asthma. And to their credit, the authors then decided to do something experimental. And basically what they did is they identified the taxa that were missing, so the specific type of bugs that were missing. And they did an experiment with asthma-prone mice that were provided either with the microbiota of asthma-prone kids or that same microbiota with the missing taxa replaced. And the bottom line of that experiment that Mary Claire Arietta put together was that much of the harm from the asthma stimulus, much of the inflammation could be relieved if the microbiota were replaced with those bugs. So that was an interesting preliminary experiment, beginning to point to a mechanism behind the long-standing hygiene hypothesis. So put this way, and this is so as not to freak out people who are into more traditional views of asthma. Anybody can have a genetic or epigenetic predisposition towards asthma. We're positing that things like C-section or antibiotics will disrupt the normal development of a diverse microbiome in infancy. And then what is known from many other studies in the literature are that members of that new microbiome are responsible for secreting metabolites, particularly short chain fatty acids, that are known to skew experimental animals and humans away from atopic responses. So if you're missing them, you're a little bit more likely to have an atopic response. There is still plenty of room for pollution, pollen, dust mites, all of the things we've been studying for decades to act as allergens and irritants and triggers. It's just that the probability of them setting you off might be higher if you're predisposed to atopic responses. And that leads to the usual thing, clinical expression of asthma. And for those of you who follow the immunology, there's similar immunology involved with hay fever or allergic rhinitis or with atopic eczema, and that needs to be explored too. So our study that we published in Lancet Respiratory Medicine in the spring brought together 3 different kinds of investigation to explore our core hypotheses. Our first hypothesis was that reduced exposure of infants, so as children under 1 year, to antibiotics should be associated with a decrease in asthma incidence at population level, and should predict asthma rates by local health area after adjustment for covariates. So that's the part we worked on at BC Centre for Disease Control. And those of you who are study hawks will say, okay, that's an ecological analysis. Exactly. So next then we said, then if there's a causal association found, that relationship will have to be demonstrated at the individual level within a prospective birth cohort after adjustment for covariates. And so that's where Stuart Turvey and crew came in with the child study. And then we also said that an exploration of changes in the gut microbiota within the cohort should support a plausible biological pathway for such an effect. So that too I'll report upon. So our methods, first of all, to the population level study. If you're in BC, you know all about BC PharmaNet. It's our database that captures all outpatient prescriptions, and it's population based and has been collecting those since about 1996. So that allows us to look at a very granular level at age and sex specific prescribing rates. So instead of just looking at overall population rates or rates in children, we can talk about rates of prescribing in children under the age of 12 months, for example. And equally important epidemiologically, if you're talking about the importance of an exposure, is the idea that we can also describe the proportion of those infants that were exposed to 1 or more course of antibiotics. So we don't just talk about the prescribing rate, we talk about that proportion. In terms of asthma incidence, some of you may know that there is a sort of a chronic disease registry project across Canada with a number of reasonably well-validated case definitions. Aggregate asthma incidence was obtained from the BC contribution to that. We have a chronic disease registry. It's based on standard diagnostic codes, ICD-9 and ICD-10. And is also enriched by pharmaceutical data on drugs that are fairly specific to asthma, such as bronchodilators. I'll talk about the reasons why we wanted to look at other explanatory variables too, but we were able to collect fine geographic data on air pollution from the CANOE registry, which collects that for BC as well. And so here is, just an example of the data from the registry. And it shows actually that when you begin to look at incidence overall, age standardized incidence, new cases of asthma are beginning to fall. But we're really interested in this specific cohort of kids. The other thing, I won't trouble you too much with the equation, but if you previously met the concept of population attributable risk, it's basically if you've got a risk factor and you take that away, what proportion of disease do you expect to disappear? And you can calculate that if you know 2 things that we can know. One is this proportion exposed. So I mentioned we can get that from the PharmaNet. The other is if you know the relative risk associated with that exposure, and we can get that from the literature and from our own studies along the way. So the association between antibiotic prescribing rate and asthma incidence, we wanted to explore at the local health area level. And in British Columbia, we've got 90 plus local health areas. And we wanted to see if we could predict asthma rate in children aged 1 to 5 by understanding the exposure of those kids to antibiotics in infancy, and also adjusting for everything else that we could. So that we did by multivariable Poisson regression. So here's results on that. So I'll give you these results on this part, and then we'll talk about the methods in the cohort. So the main thing to look at is this first figure. And the red line here is what was going on with asthma in BC. And the first thing that stood out when we began to work with the chronic disease epidemiology people is that that rate has fallen 26%. From 27 to 20 cases per 1,000 population per year. Now that means in BC's birth cohort that we see actually 1,264 fewer cases in kids 1 to 4 every year than we used to. So if you scale that to Europe, that would be 100,000 fewer cases, and nearly as many if you scaled it to the United States. The blue line here is taking a look at the prescription rate per 1,000 kids, and the axis is over on the left-hand side. And you can see that it's fallen fairly dramatically from about 1,250 to 550 or so prescriptions per 1,000 babies per day. So that's a big drop in exposure. And if you look at the proportion of kids exposed, it drops from about 65 to below 40% of infants exposed to any antibiotic. So that makes it interesting. You know, okay, the exposure's really dropped off. Is that going to have any kind of effect? Now, this is the population attributable risk thing, and I'm expressing it in terms of changes in incidence that you would predict. So 27 is where we started at the beginning of the time series, and 20 is where we actually ended up at the end of the time series we've been studying in 2014. If we take relative risks from the literature of about 1.5 or 2.0 and plug them into that equation I put on the slide a couple ago, this is what you'd expect. If relative risk is 1.5, we would have expected things to fall to 24. If it's 2, we would have expected things to fall to 23. And it's actually fallen further, which leaves room to say that either our estimate of the relative risk associated with antibiotics for asthma is an underestimate and we've missed it, or that there's more things going on at once. There are other things happening in the environment. And I'm happy if either of those is an explanation. So this is the regression. Remember, we said we wanted to see if we could predict asthma rates in young children based on the exposure of those cohorts to antibiotics. And here is a forest plot that basically says, yeah, for every increase, 10% absolute increase in the proportion of kids who are exposed, we see a 24% increase in the rate of asthma in that local health area, adjusted for other things. And because the rate of prescription by local health area ranged from about 5 in the lowest prescribing to over 50 in the highest prescribing, you can then multiply that. So you could easily be looking at, in the higher prescribing health areas, a relative risk of 2 compared to the lowest prescribing. There was a little bit of a trend over time. Young males are more prone to asthma, so it's not surprising that that stayed in the model. For those of you who are environmental people, the measures of environmental pollution, the PM 2.5, still mattered. With for every interquartile increase in that, you saw a 16% increase in asthma rate. And then some other social factors that we were able to measure at local health area level. Deprivation indices did not add a great deal to the prediction. So that's interesting. It means that at ecological level, we're seeing something. But the very first thing you should say is, okay, there are alternative explanations for ecological observations, and you can't assume that they're causal. What if there was reduction in other risk factors that we didn't measure, confounders like domestic tobacco smoke, because we know that's been dropping off to a certain extent. What if we have reverse causation? In other words, what if declining asthma is resulting in fewer antibiotics being prescribed? In nervous healthcare providers get a wheezy kid and they're more likely to give that kid antibiotics than not. So you have to worry about fixing the reverse causation issue. And what about confounding by indication? In other words, what if the respiratory infections that drive antibiotic use themselves cause asthma? So we have to sort these things out, which is why we go on to part 2 of the paper and the second study. So some of you may be familiar with the Canadian Healthy Infant Longitudinal Development Study. It's a prospective cohort in Vancouver, Edmonton, Winnipeg, and Toronto. So it's in 4 large Canadian centers that's aimed at predicting atopic responses in children to try and crack the asthma problem. And it's quite interesting because women are enrolled before they give birth. They're followed through pregnancy, birth. They and their baby are followed up at 3 months, 12 months, 5 years. And these nappies at 3 months and 12 months actually mean that a lot of these kids gave us poop, which is gold for this kind of investigation. Meanwhile, we were collecting risk factors at the individual level. Exposure to smoke, exposure to pets, ethnicity, parental atopic predispositions, and at the environmental level, we were getting the same kind of environmental measures that we were able to look at ecologically in BC. And so those are all the exposures. And then physician diagnosed asthma by the age of 5 is the outcome in the child study. So what basically we have is about 3,400 kids enrolled overall. At 3 years, there was just over 3,000, of whom 2,738 had a good clinical assessment at 3 years. And we actually did the analysis at 3 years, but our main assessment was at 5, where a full clinical assessment was available on 2644. And to note, there was a sizable subset, but it definitely was not all the kids who had poop. And that was nearly 1,000 kids basically had poop, and we were able to look at it at 3 months and at 1 year. So what did we find in the child cohort study? The first thing we found is that after adjusting for covariates, the adjusted odds ratio for antibiotic exposure before age 1 was higher than we expected. It was 2.5 in this study with confidence intervals you see there, 1.7 to 3.8. But we decided then to address the risk of confounding by indication. So we excluded the kids who received antibiotics for respiratory symptoms. And this is the forest plot that I'm going to sort of demonstrate to you here. The first thing to note is that with adjustment for everything else, and after removing kids who had antibiotics for respiratory symptoms, we still had an adjusted odds ratio of 2.15, so double the risk. We were able to deal with other important factors that are known to deal with things. Ethnicity may or may not predispose. You'll note that cesarean section without labor is known to be a risk factor. It still factors in here, but doesn't change the conclusion about the antibiotic exposure. We can see that parental atopy in the history for father or mother also is there. And we've adjusted for all sorts of things here: seasonality at birth, air pollution, tobacco smoke in the home, and so forth. So all of that, and we still see that 2.15 odds ratio. We went a little bit further and looked for dose response. And so if you are a kid with no exposure to antibiotics, you run in this cohort about a 5% risk of having asthma diagnosed by the age of 5. It goes up with exposure to 1 dose to 8.1%, with 2 doses to 10.2%, and with 3 or more doses to 17.6% risk. So there's a dose response relationship to what we're observing. So next I want to turn to the gut microbiome. I haven't gone into excruciating detail about the nature of the 16S sequencing and work. And if some of you want to know more of that in detail, I'll get you in touch with Hind, who can provide a lot more, but I can talk you through these. The first thing is that we expect over the course of the first year of life, an increase in diversity in the gut microbiome. And that's definitely shown by these two measures of diversity, PD and Chow-1. So you can see that we've got a certain amount of diversity measure here at 3 months, that it climbs quite a bit by 1 year, by whichever one of these indices are applied to the sequencing data. What about the asthma question, though? And what you can see here is that the kids who go on to have asthma by age 5 have less diversity in their gut at 1 year than those kids who do not go on to get asthma. And it's robust to both of these measures of diversity. So there is an association between a less diverse gut microbiota and the development of asthma. The next slide, though, is a little bit more challenging. Oh no, hang on. This one isn't. I'll tell you the other thing we're looking at. So this is back to the same forest plot, but we've taken out the antibiotics and we've used diversity in its place. And although this seems to be a small relationship, now we're on the negative side of 1. So as we increase the Chow measure, an interquartile increase in the Chow measure, for every interquartile increase, we see a 32% reduction in the risk of getting asthma. So that's a adjusted odds ratio of 0.68, and that is significant. So another way of expressing this thing is that diversity matters in your gut microbiota for in terms of a non-associate in terms of avoiding asthma. This is again robust for ethnicity, all those other risk factors, the air pollution, the smoking in the house, and all of those other things that we know are important in asthma epidemiology. Now, when I first take a look at this, I say, okay, maybe there's a problem with this hypothesis, because in terms of diversity, if we take a look at kids with no antibiotics and kids who got antibiotics, we're not seeing, maybe in the second measure a little bit, but we're not seeing a statistically significant difference in overall diversity. But then it becomes important to talk about other things, about the dose response and about the timing. This plot begins to look at dose response, and according to both measures of diversity, there is a significant dose response issue. So you're more likely to lack diversity if you've got more than 3 antibiotics. So the overall trend there is significant in terms of difference. But the most compelling thing we observed, and this goes along with previous clinical observation and observation in other studies about the importance of the first 3 months of life, is that antibiotic receipt in that first 3 months, 2 days to 3 months, is more likely to be associated with less diversity in the gut than at any other time. So when we sort of throw all those things into a model, we can see that antibiotic use by age 1 is significant. We see this important effect of the timing at 2 days to 3 months. We still have something of a dose response, and again, this is with adjustment for all the other factors. So then you want to go into the question of saying, for the kids who got asthma compared to the others, and for the kids who got antibiotics compared to the others, what is going on with the specific bugs that are in their gut? And so these measures here are measures of microbial abundance, basically. And if you've got an overlap here using these 2 methods, the DSeq2 and the EDGE-R, you can see these 15 organisms in the overlap group that are sort of differentially expressed by both methods. You can do the same thing with kids who get antibiotics compared to kids who don't, and you can see, okay, we've got this group of 24 here that are overlapping by both methods. But what we're really interested in seeing, those that are clearly differentially expressed with the antibiotic exposure, and in the kids who have asthma. And so that's the 6 taxa I want to just focus on the right hand of the page here. Now, most of us aren't going to recognize these particular bugs because they're not pathogens for the most part, and we don't meet them in infectious disease very often. But these are the usual suspects, these firmicute species that help produce the short chain fatty acids that have that known relationship with skewing us away from atopic responses. There's also one upregulated organism, which interestingly enough is Clostridium perfringens. Not causing disease, but it's obviously, anyway, probably not doing anything too healthy for us. So then the question arises, you've seen this association between getting antibiotics and getting asthma, and we have an idea about what's going on in the gut. Is it more likely that those antibiotics are having a direct effect on the lungs and lung responses, or that is somehow mediated through the gut? So you can actually perform structural equational modeling to explore that, where you factor in all of the covariates, the smoking, the pollution, all of those other sorts of factors, but you also factor in the diversity measures and the antibiotic modified taxa in the gut. Or you can model the potential direct effect. And the bottom line with this, which is maybe just one extra mathematical step that our postdoc brought in, was that the indirect effect is statistically far more likely than the direct effect. The direct effect is not statistically significant. The indirect effect mediated through the gut is. So I'd like to go back to a mid-20th century British epidemiologist, Bradford-Hill. Some of you, if you've done epidemiology or critical thinking, will have read about the Bradford-Hill criteria. In fact, if you're into clinical trials, he was one of the people who helped put clinical trials on the map in evidence-based medicine as well. But he recognized that not everything could easily go to a clinical trial. And so he developed this list of criteria for causation from observational studies. And the first thing he'd say is, what's the strength of association? And so we do have a pretty strong correlation in the population-wide ecological study. We also have a relative risk of over 2 in a prospective cohort study. So that's okay, it's pretty strong association. Is this consistently found? Well, it's consistent with observed associations in the literature. It also, the effects are consistent between the study designs that we were able to apply in this paper. Is it specific? Well, so far it's specific to asthma, but in fact we want to look at it with respect to other atopic diseases that follow a similar immunopathogenesis, so the allergic rhinitis or atopic eczema. Temporality, yeah, well, cause clearly precedes effect in the prospective cohort study, so that helped us get around the reverse causation problem. But it was also notable ecologically that the decline in antibiotic use preceded the decline in asthma incidence, as you'd expect for a cohort effect in the population. Was there a biological gradient? Sure, we've seen dose response in the prospective cohort study. Is there biological plausibility? We like to think that there's at least a mechanism by which this could work, which is in tune with the more long-lived hygiene hypothesis, but I think more scientific because it's measurable. Is it coherent with other information? It's certainly coherent with the other literature and other studies. Do we have an experiment? Well, not in humans. We don't have a randomized controlled trial, but I do credit Ariada and Finley for doing their mouse model experiment, which showed experimentally that this hypothesis may have some legs. And we'll actually talk about whether we can do more to prove this in a human population. And also, Hill likes us to reason by analogy. And if you're involved in other areas of therapeutics, you'll know that metabolites from the microbiome actually bring about immunomodulation in other settings and may enhance the risk of graft versus host disease and autoimmune disease under other circumstances in medicine. So what we concluded at the end of the paper, we certainly didn't talk about a certainty because we realize we're into some pretty new ground here. We concluded that a significant fraction of the decline in asthma incidence observed over the last two decades may be an unexpected dividend of prudent antibiotic use. So where do we go? What kind of things come next? Well, we certainly want to repeat these observations in other populations. So we're reaching out around the world to see if the ecological associations are also being observed, but also to other prospective cohort studies. And that's going to be important. We also think that, you know, a cohort of 3,500 kids is all well and good, but if we want to talk this as about, you know, as a population effect, we really need to do a full population retrospective cohort study using administrative data to see if this is robustly present across an entire population. We've got that underway in British Columbia right now. It's interesting to talk about future intervention trials. I don't think it will prove ethical to randomize a kid who needs antibiotics to get them or not. There is thinking that if a kid needs antibiotics, should there be a trial of mitigation of risk of asthma through reconstitution of the microbiota? That's one possibility. But there is a question on a different sort of scale that maybe an ethical experiment might be feasible, and I'd be interested in what people have to think about this. That we were discussing this before the pandemic with Gregory Fox at U Sydney, who is a respirologist, refreshingly, and is doing cluster randomized controlled trials on community stewardship in large population segments in Vietnam, 40 or 50,000 people per segment. And just before COVID hit and they had to put things on hold, we were talking about whether the outcomes he's looking at could go beyond community utilization to granular community utilization by age group and an ascertainment of asthma incidence in kids age 1 to 4 at least, if not a little longer. That would make follow-up a lot bigger. For those of you who are interested in the sort of work that we'll be doing here, I do want to stop for questions, but I will point out that Hannah Lishman is a postdoc who's working with me, and we've basically got a retrospective cohort happening where, as an exposure, we're interested in that antibiotic exposure. As an outcome, we're interested in asthma, but also other atopic disease. And we're looking at a whole host of maternal and paternal factors, kiddie factors, and other things going on in the environment to correct for other confounders. We're using the publicly available data sources in BC, Perinatal Services BC, PharmaNet, all of these other ones. And we are going to tackle the big criticisms of these kinds of observations very deliberately. Reverse causation will only include kids getting antibiotic exposure in the first year of life before any atopic outcomes. So we'll be very careful about that one. And confounding by indication, we can go pretty far, because what we can do is take a look at antibiotic treatment that's for non-respiratory illnesses and atopic outcomes. And we can also look at respiratory illnesses that were treated with and without antibiotics. So that should give us a robust sense of whether we can overcome confounding by indication. And we'll be able to come up with some measures of breastfeeding as well. So the bottom line for us too is, as those of us who are involved in stewardship programs look at this, we're beginning to say, you know, community stewardship programs were probably already a good idea for most jurisdictions in the world, because they reduce unnecessary antibiotic use. They help prevent associated adverse effects. They help reduce the selective pressure for resistant organisms. And we've shown in other work that they actually are very cost effective and save money. It's a cost saving sort of idea. If on top of that, it becomes clear that reducing antibiotic use in early life also reduces the burden of a chronic disease in the population, then I think the investment in community stewardship for most jurisdictions would have to be an unassailable priority. So the only thing that we'd want to make sure of is that we're doing no harm. And as we drive antibiotic rates down, we also want to look at unintended consequences of that, just as we looked at unintended consequences of examining the COVID pandemic. So we are taking a look at consequences like pneumonia, mastoiditis, peritonsillar abscess in kids who are not treated. And so far, we've not seen any signals in British Columbia of excessive bacterial pathology resulting from non-treatment of minor childhood conditions. But I think it behooves us to be careful about that and make sure that we haven't pushed things too far. With that, I'd like to stop and take questions. And I'm happy also to stop sharing my screen if it helps you guys so that we can see each other. But I'll ask our organizers whether I should keep things on screen in case people want me to refer to a specific slide.
Thank you, Dr. Patrick. It's Aaron. Maybe keep your screen up so that in case we need to refer to a slide. But I wanted to say thank you on behalf of all the participants for a wonderful presentation. You're a great speaker and the content is extremely interesting. So we're glad we had you come and present today. A reminder to give you a chance to take a breather, Dr. Patrick, I'll remind everyone that please place your comments and questions in the chat and I'll relay them to Dr. Patrick. So we have a few questions. The first question I'll mention is someone saying, thank you for the excellent presentation. You present a compelling scientific argument for microbial diversity. Do you have any concerns that changes in microbial diversity in the average Canadian household with children related to the pandemic measures, using more natural cleaners and bleach, Lysol, cleaner environments might cause a resurgence of asthma in the future? Could that be countered by decreased antibiotic prescriptions due to decreased viral infections?
Well, the interesting thing, thank you for that question, is that both of those things are going on. You're right. It's quite possible that kids are getting less exposure to the environment, and babies certainly do pick up these important taxa from the floor, their pets, their mom's breast, and other things, just from being messy. So there is a possibility that through less roaming, they may be getting less early exposure to these important bacteria. At the same time, we have been exploring from the point of view of the community stewardship program, the trends in use of antibiotics during the pandemic. And certainly during the first 6 months of the pandemic, as I think everyone expects, the rate of prescribing over population in BC has fallen and well below historic expectation. That may well help counter things, but I think we have to look at that in a granular way and see if that's happening in all age groups. What we don't know about that fall in antibiotics is whether it's just because people aren't having encounters with prescribers, or rather, I rather doubt that the rate of prescribing per encounter has changed, but we need to explore that a little bit.
Okay, thank you. The next question, are there other reasons that lead to less diverse microbiomes other than antibiotic use?
Well, I mean, across the human body, there's all kinds of things. So what we have seen clearly in the literature is that cesarean section, particularly cesarean section without labor, means the kid is exposed to less of the microbiota they would have normally got through transit in the vagina. And so they're behind there. Kids who don't breastfeed miss particular parts of the microbiota associated with the areola and the nipple, and that flourish in the presence of human breast milk. So those are known risk factors in infants for less diverse microbiota. In other patients, antibiotic receipt will affect microbiota in any of our, not just in our gut, but in our vagina, in our mouth, in our throat. Similarly, activities that might disrupt the microbiota could include douching in the vagina, and that may cause an imbalance, which could cause a yeast or overgrowth or a bacterial vaginosis, for example. So yeah, there's all sorts of things that mess with microbiota, but antibiotics have certainly an interesting one because their rate of use certainly coincided almost precisely with the observed asthma epidemic in the late 20th century.
Okay, thank you. A nice comment for you, Dr. Patrick. Someone described you as clear, transparent, and friendly. A wonderful speaker, so I would echo that sentiment. Another question. That you showed a couple of forest plots and there are multiple different analyses. In this kind of study, how do you control for doing multiple tests and seeing things that may not be real just by multiple testing?
That was implicit in the regression that informed the forest plot. So
Okay.
it, if you were doing multiple univariable tests, you'd have to adjust your p-value. That's essentially done within the regression calculation itself. So if I show you a statistically significant result in that association, that's net of the number of variables that are in the model, and it's also adjusted for the effect of other variables that are in the model.
Okay, great. And I have a question for you. You had referred to other studies showing similar signals or associations. Are there many studies? Do they show similar associations in some cases, but differences in other ways? What's your overall sense of the evidence?
Well, my overall sense of the observational evidence is that you sometimes see more aggressive associations described in cross-sectional studies than in prospective cohort studies. So I tend to put more weight on the prospective cohort studies. So that's that's a key one. But what I'm interested in are these studies that can adjust for multiple confounders in the home, in the maternal, paternal, and child experience. And there are probably 3 or 4 other studies globally that have the ability to take a look at this. So we're interested in this study in state of Victoria in Australia. It's a community actually west of Melbourne, but we're trying to get in touch with those investigators to see if the same kind of analysis is possible there.
Okay. Okay, another question. How was the, 2 questions. How was the diagnosis of asthma made? And is there any relationship with decreased microbial diversity in chronic bowel conditions like irritable bowel and inflammatory bowel disease?
Okay, so those are 2 different questions. So this was physician diagnosed asthma, and I think it is a really good question because in little kids, that's going to be, the getting perfect compliance with things like pulmonary function tests is not the same as when you're trying to diagnose an adult or an older child with asthma. It's the best standard that the originators of the cohort study could come up with, and we certainly are looking to see if this same cohort of kids is exhibiting similar effects as they age, and as they age, then they also come online with the highly objective pulmonary function tests or stimulation tests, and that will help us a little bit further. So I think one reason why it's been hard to take a look at this before, well, there's a couple of reasons. One, people tend not to look at antibiotic use in a very granular way by age. And the same thing with asthma. We haven't taken a very granular look with asthma. So it really took studies that were specifically focused on childhood asthma and its incidence and the factors associated with it before it was really possible to see some of these associations. Okay.
Another question, when you talk about exposure of infants to antibiotics, would that include the mom receiving antibiotics and then the baby being exposed through breast milk?
You can address those things in a study in different ways. At the population level in BC, we were only in our initial study able to look at antibiotics given directly to the babies. So that's the ecological study. In the child study, we were able to look at both. Basically, antibiotics given to the infant seemed to be the most important here, and that actually would make sense because a large proportion of that antibiotic dose gets to the gut and has a direct effect there.
Okay, and another question related to probiotics. Can you comment on the role of probiotics in infants and/or adults with and without
I would say
antibiotic
this
exposure?
about probiotics. To date, as most of you know, probiotics have not been very intelligently designed. They've been relatively benign flora that we know how to grow and that grow easy in vats. But the design of probiotics has not been informed by a big understanding of the function of different taxa in human health. What's happening now in the sequencing era is we're gradually getting a better understanding of that. And so people are deliberately designing probiotics that might replace more critical taxa with known function. It's still not a slam dunk that those will be great therapeutics, because you have to be able to get them to the site where they need to grow, and you have to assure that they're able to grow in that population and not get out competed by something else. But current probiotics that are on the shelves, with the exception of one or two that have undergone clinical trials, they're pretty well a placebo.
Okay. Great. Just give you a break for a second and remind the audience that if you signed in late, we had mentioned the continuing education credits that are available to you. After the session, within the near future, you'll receive an email with an evaluation form. If you complete the evaluation form and send it back to us, within a little while, you'll receive your continuing education certificate. If you're not interested in the credits, we'd still like you to complete the evaluation form, because we do go over them carefully in an attempt to try to make this webinar series better. So any comments you have to submit through the form would be welcome.
There was another question about other association of microflora problems with other conditions, and I didn't get to that. That was the second question from one of our audience. And microbiota changes are terribly important. Everything from bacterial vaginosis, which we've understood for a long time to be related to depletion of lactobacilli in the vagina, to the suspicion that they may have to do with predispositions to obesity and other conditions. But it's an active area of inquiry over time. I think one of the most fascinating areas is that of brain health, where there's active exploration of whether signals from gut microbiota affect certain degenerative brain conditions. But I would say that the jury's still out on a lot of that, and we need more investigation, you know, like we say, Bradford-Hill criteria plus some good experiment and clinical trials before we're really dead sure that we've got the real McCoy.
Sure. Okay. You had mentioned that critical time period, 2 days to 3 months. Is that time period also been something that's seen in the other studies?
Yes. So other studies that have taken a look at infant exposure to antibiotics have commented on the apparent importance of the 3 months. And then certainly in animal models and so forth, the very early postnatal period is important in terms of colonization and its disruption. Okay.
Another question. You had mentioned the rate of antibiotic exposure ranges from a few percent to 50% in different jurisdictions. So I'm curious, where do we fall in terms of that, and what's going on in the places that have 50% exposure?
So this is across British Columbia. So as you might imagine, that at local health area, like small community level, there's a lot of variation. And in some communities that have a very low population, all you need is 1 or 2 outlying prescribers to really send the rate south. In terms of antibiotic use over population, British Columbia leads Canada in having the lowest rate over population overall. We are not as exemplary as the Dutch or Danish, and we tend to run a little lower than the American average. And of course, the antibiotic use across the US is extremely diverse, with very high use in some parts of the country, much lower use in others, and it almost follows the electoral map.
Interesting. Okay, we're approaching 8 o'clock, so maybe we'll finish with a couple more questions. One is, can you please mention the names of the 1 or 2 probiotics that have been studied and what conditions have they been studied in?
Well, I don't use them regularly or write scripts for them, so that's my problem. But there, and maybe Tom can help me here. But there was one that was found a bit useful for irritable bowel in one randomized trial, BV3 or whatever. But I usually try everything else before I send somebody to get a specific probiotic.
Okay, and then here a comment, BSL3 maybe is the name. And I guess one other question here is, would you suspect that some antibiotics are worse than others for this?
Absolutely. And the problem with our study was, with kids is there's not a lot of variety in what these kids get. As you can well imagine, the vast majority of the kids who got antibiotics got amoxicillin or a variant. And so we weren't really able in this study to make a significant comparison between classes. But that's not the case when it comes down to other conditions. I mean, probably the most well-known association is that if you use, penicillin for oral indications in dentistry, you have a much lower risk of getting C. difficile than if you choose to use clindamycin. And the take-home story there is you better be sure you've got a penicillin allergy, and you really want to test that and delabel your patients as having beta-lactam allergies if they don't have them. So that's pretty critical. The other thing that's been observed is that you're much more likely to run into problems in the gut if you really clobber the anaerobes generally. So it's not just clindamycin, but it's any antibiotic that takes out the anaerobes. It seems like the more protective bacteria in the gut were ones that most of us never learned about in medical school because they're hard to grow in culture, but they're basically nice benign anaerobic flora that float around in there, do good things for us metabolically. And when we clear them out, C. diff is more likely to be a problem. And there's also an interesting connection with graft versus host disease in people going through transplants.
Okay, thank you for that. Well, it's 8:01 now. I want to thank you again on behalf of the Therapeutics Initiative and all the people that called in to participate, and also thank the audience for taking time out of your evening to spend this with Dr. Patrick and us. And we wish you all a pleasant evening and hope to see you at our next webinar on February 10th.
Back at you. Thanks very much for having me, and good night, everyone.
Good night.